Hello, this is the video for Lab 9, Special Senses. If you go to page 95, you'll see that there's a list of accessory structures, A through D. You can scratch all of those out. They're not on any of our models. And then if you go over to page 96 for the ear, if you go down to 3, we're going to call it the Bony Labyrinth rather than the Osseous Labyrinth. And you can cross out utricle and saccule underneath vestibule. And spiral organ is spiral organ, so you can cross out organ of Corti. So this is the eyeball. The outer layer of the eyeball is composed of the cornea, a transparent structure on the anterior side of the eye that light passes through. Then the bulk of the outer eye is composed of the sclera, a dense opaque white structure that gives the eye its shape. And then at the posterior end is the optic nerve, carrying the information to the brain. The middle layer of the eyeball is made up of the choroid coat. This is a vascular structure, so lots of blood vessels being brought to the eye through the choroid coat. At the anterior end of the choroid coat, it thickens into a muscular structure called the ciliary body. This forms a ring, a muscular ring around the lens. Here's the lens. And then there is a space called the anterior chamber behind the cornea that is filled with aqueous humor, with fluid. The lens is connected to the ciliary body by these thin structures called suspensory ligaments. This allows the ciliary body to control the shape of the lens to focus light to the back of the eye. And then we also have this ringed structure that's nice and colorful called the iris. It's made up of smooth muscle tissue, and it's able to control the size of the pupil. The pupil is a hole that allows light to pass through and into the eye. So light will go through the cornea, through the pupil, through the lens, and to the back of the eye. The innermost layer is the retina. So most of this here is the retina. The retina is where we have the photoreceptors. It is what detects the light that is arriving into the eye. Directly across from the pupil is a darker structure called the fovea centralis. This is where we get the absolute best vision possible, and it only has cones, photoreceptors cones within it. Also at the back of the eye is a lightish colored structure called the optic disc. This is where the optic nerve is forming off of the eyeball. The optic disc is also known as the blind spot because there are no photoreceptors here. So we actually have two little spots in our field of vision that are actually blank, but our brain fills in the blanks. Also in the back of the eye is this large area that is the posterior cavity, and it is filled with a gelatinous substance called the vitreous humor. So that is the parts of the eyeball. Various tests, vision tests that are out there. We have the visual acuity test. This is for seeing how well you can see things at a distance. We have the blind spot test. This is actually on page 99 of your lab. You can use this to actually detect your blind spots. There's the color vision test to see if someone is colorblind or not. There's the astigmatism chart. This is for seeing if someone has astigmatism, that is irregularities on the curvature of the cornea and lens. And there's also the accommodation activity. This is actually testing for nearsights, for your near vision, how well you can see things that are close to your face. These are some various tests used for people's vision. You should be able to be somewhat familiar with their names and what they test for. Here is the overview cartoon of the ear. The ear is broken into three areas: the outer ear, the middle ear, and the inner ear. The outer ear is composed of the auricle, this big fleshy part that you can get pierced if you're so desiring. It funnels sound waves into the external acoustic meatus, that is air field, also known as the ear canal. And then those sound waves hit the tympanic membrane or eardrum. This then transmits the vibration into the middle ear. The middle ear is also an air-filled chamber that connects to the back of the throat through the auditory tube. This allows us to equalize pressure over the tympanic membrane. The vibration of the tympanic membrane gets transmitted to the inner ear through the auditory ossicles. These are the ear bones. Their names are malleus, incus, and stapes. So the tympanic membrane connects to the malleus, malleus connects to the incus, incus connects to the stapes, which connects to the oval window of the inner ear. So when tympanic membrane vibrates, it causes the malleus to vibrate, causes the incus to vibrate, causes the stapes to vibrate, moves the vibration into the inner ear. Here's just that cartoon again with those model bones being detailed. This area here is the inner ear. It is a bony structure, so it is called the bony labyrinth. Coming off the bony labyrinth is the vestibulocochlear nerve that carries the signals of hearing and equilibrium to the brain. The bony labyrinth can be broken up into three regions. You have the semicircular canals that are three loops. You have the vestibule, which is a blocky area that has the oval window and round window, and you have the cochlea, the Snell shell, where we detect hearing. The semicircular canals have widened areas. These widened areas are called the ampulla. So semicircular canals have ampulla. The vestibule, as I mentioned, has the oval window, which is underneath stapes, and the round window. Here's an image from our model. Semicircular canals with their ampulla, vestibule with the oval window, cochlea. There's the vestibulocochlear nerve. If you open up the cochlea, you see it has chambers such as the scala vestibuli and the scala tympani. These are fluid-filled. Turns out the vibrations from the stapes goes through the oval window and into the upper chamber, the scala vestibuli. When we talk about these chambers, we will actually use this model. This is a cross-section of the cochlea. So it is a cross-section of one region of this structure. So within the cochlea, we have the upper chamber, the scala vestibuli, filled with fluid called perilymph. We have the cochlear duct, also filled with fluid called endolymph, and the scala tympani, filled with fluid called the perilymph. The vestibular membrane separates the scala vestibuli from the cochlear duct. The basilar membrane separates the cochlear duct from the scala tympani. This entire structure is the spiral organ that detects hearing. Take a close-up look at the spiral organ. This piece here, this piece of cartilage, is the tectoral membrane, and underneath it is a bed of cells, including the hair cells. Hair cells have little hairs on them. The hairs are embedded in the tectoral membrane. The cells here rest on the basilar membrane, so that when pressure waves from the sound waves, from the vibrations arrive, they cause the tectoral membrane to vibrate, which bend the hairs on the hair cells, and that generates the signal that gets sent by sensory neurons to the brain. So hair cells, the receptors are mechanoreceptors. Change in shape is what they detect. So again, here is the model. This represents the tectoral membrane, the cartilaginous structure. This is the spiral organ, and you can see the little bitty hair cells with the little hair bundles on them. So, sound waves arrive through the external acoustic meatus. They hit the tympanic membrane. Tympanic membrane vibrates, causing the malleus to vibrate, causing the incus to vibrate, causing the stapes to vibrate, causing the vibration to be passed through the oval window into the scala vestibuli. Vibration is now pressure waves traveling through the fluid. It'll go along the length of the cochlea till it reaches the area that is attuned to that frequency of vibrations. It'll pass into the cochlear duct, hit the tectoral membrane, causing the hair cells to bend, giving us a signal for hearing. Any remaining pressure waves will then enter the scala tympani, travel through it to the round window to be dissipated into the middle ear. This way we avoid any feedback. There are two tests for detecting hearing loss, Rhine and Weber. Basically, the point is that if you hit a tuning fork, it makes sound, and if you can not hear it through the air by holding it near your ear, then that means you have hearing loss. If you hit the vibrating tuning fork and hold it against the bone on your skull, here or here, then if you can hear that sound, then your nerves are fine. The damage is to the outer or middle ear structures. If you cannot hear the sound when it's being transmitted through the bone, then that means you have some sort of sensory damage. Something's wrong with the nerves carrying the signal to the brain. And that is it for this lecture. Be sure to complete all the activities for this lab.